also continuing on the topic of mass spectrometry, and in particular phosphoproteomics, and Pengyi's lab is responsible for the PhosR Bioconductor package, which includes steps such as imputation, normalization, and functional analysis of this data. So yeah, when you're right, Pengyi, if you can share your screens. Yep, we can see those. I'll just make sure it looks okay when you go into presenting.
Hopefully I'm sharing the right screen.
That looks great. Thank you so much, Pengyi. And I'll pass it over to you.
Okay, thank you. Thanks for the opportunity to present today. I'm going to talk about PhosR, a package we developed for processing and functional analysis of phosphoproteomic data sets. So firstly, I want to introduce the PhosR package. We implemented two major components in PhosR, including processing and downstream analysis. And in the processing part, we have different diagnostic plots, and we have filtering, imputation function, as you just heard. And we also have a normalization and batch corrections as modules in the pre-processing component. And then in the downstream analysis parts, we have different enrichment analysis, and we have kinase substrate predictions, as well as functions for identify the Signalome in the phosphoproteomic data sets. So I will start with the processing aspect of the package. And I want to start with imputation. Here we use a heuristic approach to implement different imputation functions. We here will introduce a few of those imputations functions we implemented in PhosR. I will start with what we call site and condition specific imputation. And this is a function that the user can specify a quantification rate for a given phosphor site, which assuming have multiple replicates in the same condition. This quantification rate will control out of how many biological replicates, for example, you have quantification for the remaining missing values for that phosphor site to be imputed. And in this case, as an example, if we say this quantification rate is 50%, and then this example phosphor site X here will have more than 50% quantification out of the 6 biological replicates. And then the missing values here will be imputed by using the empirical normal distribution we created out of those quantified values here. So this is a way to ensure the imputation applied to this phosphor site is specific to the quantification we observe from the other quantified biological replicates. And as a contrast example here for the phosphor site Y, we have this quantification rate as greater than 50% out of the 6 biological replicates. And in this case, the phosphor site Y wouldn't fulfill this requirement, and the missing values here wouldn't be imputed using this function here. The other typical function that's used in the phosphoproteomics community is called tail imputation. And this is firstly introduced in MaxQuant by Matthias Mann's group. Essentially, what this function is doing is to use the quantified sites across the entire sample, and then create this empirical normal distribution out of those quantified sites, typically hundreds of thousands of those. And then a downshift was applied to this empirical distribution by using some fixed parameters here that can be controlled by the users. And then typically these values are set as like this. Then we will be able to impute some of those lower end values that we will be using as to capture the lower detection range of the mass spectrometry to impute the missing values. So this is another function we implementing in the PhosR package, which is the same approach that's used in MaxQuant. And thirdly, we have another function called paired tau-based imputation. So essentially, this is a function that utilizes the tau imputation, but allows the user to control a few other parameters. For example, let's say we have a basal condition and we have a stimulation condition in our phosphoproteomic data sets. And for these two conditions, we have, say, biological replicates as 6 for each. Then those are the parameters that the user can control. For example, if we require the basal condition to have completely missing out of all the 6 biological replicates, and for the stimulation, we require this to have more than 50% quantification rate, then the phosphor sign X will fulfill this requirement. And the missing values here for X, this sign X will be imputed using the tau-based imputation approach as introduced here. And this is to ensure this phosphor sign will be included in the calculation of the fault change and downstream analysis. As this type of missing is biologically meaningful in most cases. As you can see here, the basal probably correspond to condition that doesn't have much phosphorylation happening in the system for this phosphor sign. But in the stimulation condition, clearly we have consistent phosphorylation on this side in the system. So we do want to include this for downstream analysis, and without applying imputation, this side will be lost in the downstream analysis. And that was biologically motivated application to impute this phosphor site in such a manner. Just as another contrast, we have the site Y here. That doesn't fulfill the requirement we set up here, which can be changed by the user. So that's why the phosphor site here doesn't receive any imputation using this function here. So let's look at how this imputation is performing using some real data set. So we utilize this data published in this 2015 study. It has 2 liver cell lines, and it has either control or insulin stimulated conditions. And for each of these conditions, either control or insulin, for each cell line, we have 6 biological replicates. And this is the quantification rate before applying any imputation. It's around 60% quantification for each of the sample. This is the result after applying the imputation, and we use the default parameter that I introduced in the previous slide, and we have the quantification rate close to 80% after applying the imputation. And the other important observation is actually here. So this is the data before applying imputation, and we can see the biological replicates from both the first cell line and the second cell line. The 2 conditions are actually mixed together in this clustering. So that's not the perfect clustering result. And here, after the imputation, we can see clearly the biological replicates from different cell lines as well as different conditions are correctly clustered with the imputation data. So that's showing overall we have a better reproducibility, but does it actually have any impact on the downstream analysis? To answer that question, we looked into the differentially regulated phosphor site in the 2 cell lines before imputation and after imputation. So for the first cell line, as you can see, before imputation, we have about 500 downstream downregulated phosphor site and 300 upregulated phosphor sites. And after imputation, we have increased this number to 1,200 for the downregulated and 700, more than 700 for the upregulated phosphor site for the first cell line. And for the second cell line, we observe a very imbalanced result before imputation. So we have about only 80 downregulated phosphor site, but more than 1,400 upregulated phosphor site. And after imputation, we do see the result are much more balanced. So now the question is whether this has any impact on the downstream analysis, even though we have more phosphoride regulated in the imputed data compared to those unimputed for the first cell line. And for the second, we have more of a balanced result. So to really look into whether this is biologically meaningful, we take advantage of the prior knowledge. So we do know in these two liver cell lines, the cell will respond to insulin signaling. So we take advantage of those reactome data, reactome pathways that's associated with insulin signaling, such as PI3K events, as well as those regulated by PI3K and AKT activation. And since we know those are the signaling pathway that should be activated in the insulin stimulation, we can see whether the enrichment for those pathways in our imputed data improve compared to those that does not receive any imputation. And that's indeed what we observe here for both cell lines. We do see better enrichment as indicated by the y-axis here for our imputed data compared to those that does not have any imputation. So, so far, I have shown the imputation that we implemented in PhosR allows us to improve the number of quantified phosphosites, and it also helps us to improve the reproducibility of the clustering. It also allows us to identify the differentially regulated phosphosite more accurately. That leads to more meaningful downstream pathway analysis. So I want to move on and introduce the second modules in the processing aspect of the PhosR package. We implemented normalization and batch correction function based on the RUV framework. RUV stands for removal of unwanted variation. And it's a popular package, a popular framework that was used for mini microarray data analysis, and later it was extended for both bulk RNA-seq and single-cell RNA-seq data analysis. And here we use the same framework, and to be able to run IUV, one need to introduce what's called positive control, which are the phosphosite in our application that does not change in the data set. And to identify those phosphosite for using the IUV framework, we need to identify what we call stably phosphorylated sites, or SPS. So the approach we identify those stably phosphosites are to utilize the high quality data sets, phosphoproteomic data sets that has been published previously. We found 4 high quality data sets that profile different cell lines as well as using different stimulations. And we identified the overlapping phosphosite that's identified in all the 4 data sets, making an assumption that if phosphosite is identified across different cell lines, then the phosphosite is likely to be housekeeping, and in that sense, likely to be less regulated in a cell type specific manner. So that's the 1200 phosphosite we identified out of these 4 data sets. And out of these 1200 phosphosites, we rank them based on how much, what's the fold change they have in each of their respective data sets. And the idea is to find the phosphosite that has minimum regulation upon either differentiation or stimulation using chemical. And from these ranking lists, we combine the rank into a consensus list using Fisher's methods. And this generates consensus ranking list with the phosphosite that's minimally regulated or changed in the conditions out of the 4 data sets at the top of the list. And those more regulated by the treatment or differentiation at the bottom of the list. So here's a quick quantity control. The first box corresponds to the site that's uniquely identified in each of the 4 data sets. So these 4 sets are presented here for each of the 4 data sets. And then we have the overlapping sites. That's the 1200 phosphosite we identified across the 4 data sets. So that's the second box. As you can see, as what we have hypothesized, the overlapping phosphosite do show less regulation as indicated by this y-axis, the log24 change, the absolute log24 change. So we do have less regulation to those overlapping phosphosites, suggesting they are more likely to be housekeeping. And then the next 3 boxes correspond to the top of the list, the top 100, and the middle of the list here in the consensus list, and the bottom ones. And as you can see, we do have minimum regulations for the top ranked phosphosites in each of the 4 data sets. So those are the phosphosite we included in the PhosR package as our positive control or stably phosphorylated sites for normalization. So how does this function perform? We again utilize the dataset from this 2015 study to evaluate the normalization and batch correction component in the PhosR package. Here in this publication, we have 2 independent data sets. The first one is insulin, is called early insulin Signalome. Basically, this data set is generated by profiling the liver, the mouse liver, and then profile this in a time course to see how the insulin stimulation to the mouse liver respond. And then we have the second data set, it's called intermediate insulin Signalome. It is profiling this, the time course again, but in a different time scale. So out of these 2 data sets, we actually have 2 time points that's matching. So clearly the starting point, 0, and also the 30 seconds of the first data set correspond to this half-minute time point, 30 seconds in the second data set. So we can imagine combining these 2 data sets will create a more comprehensive temporal profile for this study. So what we did here is to naively just combine the data sets together and generate this clustering. And what we can see is that the 0 time point and the 30 second time point in the 2 data sets are completely separated from each other. And that's not ideal. That's not what we want. And after applying the normalization implementation I introduced, we now see the 0 time points and the 30 seconds time points from the 2 data sets mixed together, suggesting we are able to now remove the batch effect from these 2 data sets and generate a cohesive temporal profile out of all the time points we have for these 2 data sets. As another demonstration, in this study, we also generated our own phosphoproteomic data sets. What we did is we take the rat myotubes and we have 3 conditions and 1 basal. So we have 3 stimulations, ACA, insulin, and ACA plus insulin. So those are the stimulations that will create a Signalome cascade, and we want to see what are the Signalome cascade underlying those different treatments compared to the basal condition. So we use a technique that's called SILAC that label the peptide and the phosphosite, and we will use this to quantify the phosphorylation changes upon the stimulation compared to the basal condition. So in this experiment, we generate 4 biological replicates for each of the stimulation compared to the basal. While the ACA stimulation biological replicates are clustered in a single group, the insulin stimulation and the ACA plus insulin are actually confounded by the experimental design. So as you can see here, we have the experimental 1 clustered as a single group, and then experiment 2 as another group, and experiment 3 and 4 as another 2 groups. So this shows we do have experimental design batch effect in the data set before any normalization. And this can be seen on the PCA plot as well. So here is the result after applying the normalization, and we are able to correctly cluster those treatments into their biological replicates after the normalization, as indicated by this clustering here, as well as this PCA. So that's the key component in the processing that I just introduced. Now I want to move on to the downstream analysis. So the first thing I want to introduce is the typical functions. So clearly, whoever does phosphoproteomics experiment want to identify the signaling pathways. So what we have in the PhosR package is the typical pathway enrichment analysis. So here as a demonstration, we try to identify the pathways that's regulated by insulin compared to basal in this myotube phosphoproteomic datasets we generated for this study. What we have here is two ways to calculating this enrichment. The first one is the typical overrepresentation analysis using Fisher's exact method. And those are the signaling pathway from using Reactome database we put out from insulin compared to basal. As I mentioned, this type of cell myotube, they respond to insulin compared to basal, and those are the signaling pathway that's actually expected from the insulin stimulation, including mTOR and AKT signaling. We have another function, which is more like a gene cell enrichment type analysis. And this function is different from the overrepresentation analysis because it does not require you to have a cutoff for calling what are the differentially regulated phosphosites. So it will take the entire list of your phosphosites into account to calculate the enrichment for the signaling pathways. Clearly, these two approach have performs similarly, but also have different results as indicated by the pathway we put out using this different approach here. So that's the pathway enrichment analysis implemented in PhosR in a brief introduction. We do have many more functions that I couldn't get into. But I want to move on to the second aspect, which is the kinase perturbation analysis. Another key question in analyzing phosphoproteomic datasets is to identify the key kinases that's driving the signaling cascade in the system. Here we have this dataset that use different stimulation to stimulate the myotube. And here what we demonstrate as an example is to look at the difference between insulin stimulation compared to basal and ACAR stimulation compared to basal. So these are two different stimulations, and we can use this what we call two-dimensional kinase perturbation analysis to identify what's concordant and what's discordant. And as a positive control, we highlight this kinase here called PRKAA1. This is a kinase that's a subunit of AMPK, and this is a kinase that's known to be induced by ACAR stimulation. And as you can see, this kinase has a positive z-score change on the y-axis, which captured the ACAR versus basal condition. And on the x-axis, it does not show any change in the insulin stimulation. And this is demonstrating this kinase is specifically induced by ACAR, but not insulin. So as a positive control, we demonstrate this is a useful function that can help us to dissect the different kinase that's regulated by different stimulation conditions. So just to move on, I want to introduce the next function, which is the Signalome network construction function. And this function utilizes another function called kinase substrate prediction, which I wouldn't introduce today. But just to point out, this is a typical application in phosphoproteomic dataset analysis, which is to identify potential substrate for a given kinase. And we do have this recent publication that we introduced this method in more detail. By using this function in PhosR predicting for the kinase substrate, what we can do next is to use those predictions to reconstruct the potential crosstalk in the biological system. And in this case, we generate this diagram. that captures the potential crosstalk of different kinases based on the different substrates that they regulate, in which we partition this into 6 clusters based on their temporal profiles, based on their stimulation profiles. So one of the interesting questions we can answer using this data set is to ask whether we can rediscover the synergistic effect between AMPK signaling and insulin signaling. The reason we ask this question is it has been demonstrated previously from those publications that the AMPK signaling and insulin signaling work together to regulate the cell. And we are wondering, since we have this 2 treatments, whether the PhosR package will allow us to recover this synergistic effect. And at the same time, we also want to use this tool to discover whether there's anything that's unknown for those signaling, since they do trigger different signaling cascades, whether there's anything other than just synergistic effect. So the way we look into this is to use the function called Signalome map, which convert the core diagram into this string plot. And from this, we can see the 6 modules we generated in the data set, and we can see different kinases are regulating those modules. And in particular, I want to draw your attention to the PRKAA1 and 2. As I introduced, these 2 kinases are 2 different subunits of the kinase called AMPK, and is regulated by ACA signaling. And as you can see, these 2 kinases, these 2 subunits of the same kinase are regulating the module 1 and 3 and 4. And in the module 3, it looks like they are uniquely modulating this single module. And module 1 and 4 looks like those kinases are working together with other kinases to regulate this other 2 modules. So what we did is to look into the fold change of the phosphoside in those 6 modules. And now what we see is that, as has been reported previously, we do have those ACA plus insulin stimulation giving us more fold change or stronger fold change compared to their individual components, suggesting there is a synergistic effect of the ACA and insulin stimulation for module 1 as well as for module 2, module 4 observed here. However, we see interestingly for module 3, we don't, as expected, we have a strong ACA stimulation in this module, and we don't have any stimulation from insulin, but we do see the combination of ACA and insulin actually reduce the signaling for module 3, suggesting there is some kind of repression from the stimulation of insulin when you trade it together with ACA. So from this, we conclude we rediscover the previous reported synergistic effect of AMPK and insulin signaling on these 2 modules, module 1 and module 4. And interestingly, we also discover a new module 3, that's basically stimulated by ACA, and it has inhibition effect by insulin signaling. So just to summarize, PhosR is the first bioconductor package for comprehensive phosphoproteomics data analysis. And today I introduce some of the elements from the processing and downstream analysis for the PhosR package. And I want to direct people to who are interested in using our package to the PhosR Bioconductor page, as well as the STAR protocol that we publish together with this package that gives us the stepwise application procedure for using this package for your own data set. And with that, I would like to acknowledge the collaborators from Charles Perkins Center for generating the phosphoproteomic data sets that we use in this study. And I want to highlight Hanny King and Taiyuan King, who are the co-lead for this project, and Dee for contributing to this project. And I thank you for your attention and happy to answer any questions.
Thank you, Pengyi. for your talk, and thank you again for switching slots. Just a quick question from me so that we can give Mahmood time to talk. With the RUV results, looks very impressive for the normalization or batch effect correction. I've not applied it to, I've applied it to gene expression data, and one of the challenges there is in choosing how many unwanted factors to use. How do you approach it for this type of data?
Yes, we use a heuristic approach. So we will try a few values and see which one gives us a meaningful result. Normally, yeah, that's a very difficult question, and there is no good solutions to that question. So one of the suggestions is to use a large number and then gradually count down to a smaller k and see whether you can maintain the result reasonably well.
What sort of values do you typically use?
I guess the assumption is how many confounders potentially you have in the experiment. And normally I will try something like 5 and see what the result would look like. And if it still looks pretty bad, I will try something like 10 and then work from there.
Sure. If you have any other questions,
best question to your question, but best answer to your question, but that's practically what we are doing.
Yep. Thank you, Pengyi. And if you have other questions, please ask on the Slack channel. We'll finish with